Today's webinar will be about enrichment tools in the VEuPathDB resources. This is a recorded webinar in case if you're joining us midway. There are a few housekeeping things that I would like to just mention. So in terms of the webinar is being recorded, and you will receive a follow-up email with a recording link and also a e-certificate of the attendance. The notes also will be posted on our webinar page, which can be navigated to by clicking on the Help menu, then choosing Learn how to use VEuPathDB and Webinars. In terms of the general housekeeping things for the GoToMeeting, if you have any questions, please ask them within the questions section, and we will try to answer them as we go through the webinar. Today, my name is Evelina Basenko, and today I will be presenting a seminar with Stuart Brown, a VEuPathDB team member based at the University of Pennsylvania. I am also joined on the call by other VEuPathDB members, Gloria Geraldo Calderon, Suzanne Warrenfeld, who will be maintaining questions in the background and will alert us if there's anything that we need to answer within the webinar. So just a few notes before I'll pass it on to Stuart. Today we will discuss certain post searches enrichment tools that deal with gene ontologies, EC numbers, and also word enrichment. We'll touch base on in terms of how to deploy these tools using the searches embedded within the VEuPathDB resources, or how even to bring in your own lists of gene IDs. Right, Stuart, so the floor is yours.
Hello, I'm Stuart Brown with VEuPathDB. I'm going to talk about basic background of genome ontology and some of the math for enrichment analysis. The concept of gene function is very valuable, particularly when you have a group of genes that are not that well characterized, but you need to have a data set in order to apply functions to your genes. Particularly with the VEuPath genomes, we have a lot of parasites and other organisms that may not be that well genetically characterized, and experimental knowledge obtained in one organism is often applicable to others, but biology is complex.
Sorry to interrupt. Are you sharing your screen?
You're not seeing my screen?
I do not see the screen. Can others confirm what they see?
Well, it's showing, it says it's showing my screen. I don't know what else to do.
Okay.
Yeah. Perhaps unshare, then share again, because I don't see it either. I see your cursor moving around.
Oh.
Yeah. Try unsharing and sharing it again. There we go.
There you go. Yep.
Oh, well, that was weird. I didn't really do anything. I just moved the cursor around a little bit. Does it change slides? Yeah, now it's changing slides. Okay. I was saying biology is complex and functional information about genes can be difficult to transfer across organisms. A small number of genetic model organisms have been studied very extensively, often with mutations in every single gene. However, to talk about biological function, it's necessary to make an abstraction to identify the general process involved in the gene function rather than describe the phenotype in a very organism-specific member, something like pink eyes or bristles or a swimming behavior. So the Gene Ontology Consortium was specifically designed to answer this question. Some quotes from their mission statement: the GEO Consortium was developed to create an up-to-date comprehensive biological system from the molecular level to pathways and cellular organization. The GO knowledge base is the world's largest source of information on the function of genes. And this knowledge is a foundation for computational analysis for molecular biology and genetics research. And that's exactly how we want to use it, for computational analysis of function. The GO consortium began in 1998 with just 3 organisms: fruit fly, mouse, and yeast. And these model organism or geneticists worked collaboratively to develop a common classification scheme for gene function. The current GO includes model organisms from fruit fly, mouse, rat, yeast, worm base, zine base, and zebrafish. The word ontology specifically means a defined relationship of vocabulary of clearly defined terms within a system of categories and relationships. In biology, we use it to talk about a method to name biological processes uniquely, systematically, and consistently. Ontologies are generally structured as a hierarchy from more general to more specific terms. What this means is that each term can have both parents and childs, things that are higher in the hierarchy and things that are lower. This is particularly helpful in biology, where we can define the function of a protein in a stepwise fashion. For example, a membrane protein is a transporter, is an ion transporter, is a calcium transporter, is a voltage-gated calcium transporter. And other transporters would fit into this hierarchy and then branch off. An example of this, an ontology for the bioprocess glucose catabolism to pyruvate, starts at the very top, biological process, works its way down to being ever more specific. You'll also notice that the arrows go in multiple directions. In other words, one parent can have more than one child can have more than one parent. They call this an acyclic graph. What it ends up meaning is that for each particular process, there's a large number of terms that can be associated with that bioprocess at different levels of specificity. GO actually contains 3 different ontologies. The bioprocess describes sort of more general processes like energy metabolism, reaction to stimuli, DNA repair. Molecular function maps to more specific metabolic pathways, but can also be used to describe the general activity of a protein, such as catalysis or transport. But a molecular function doesn't specify where, when, or in what context the action takes place. And then the third ontology is a cellular component, which refers to a location in the cell, such as a membrane, a nucleus, mitochondria, ribosome, etc. So these 3 ontologies are completely separate. The parent-child relationships don't go across the different ontologies. And you can choose whether to apply one or the other of these 3 ontologies when you're labeling functions onto a list of genes. The information in GO is based on experimental facts, mostly gathered from the literature. So every term that's annotated onto a protein has a traceable evidence-based statement that relates a specific gene product to a specific term. So the current GO has 150,000, actually findings from 150,000 papers representing over 700,000 experimental annotations. An additional 6 million annotations are computationally inferred, generally by protein sequence similarity. So a very similar protein in a related organism could take on the same function that's been experimentally proven in a model organism, and that would be a computationally inferred function. Now, to talk about gene lists, many kinds of experiments produce a list of genes as a result. Differential gene expression, perhaps by RNA-seq, genes that are restricted to a particular clade. So you could say what genes are common among all this group of species. Transcription factor binding would be a functional analysis. Protein-protein interactions generate a list of genes. The targets of regulators, SNPs associated with a disease, or SNPs that differentiate two strains or species with different phenotypes are all experiments that can generate a list of genes as a result. Now, the question often becomes, given that list of genes in your experimental result, how to identify interesting properties of the list of genes rather than random, or rather than just cherry picking a function that you notice on the list. How do you prove that that list actually contains multiple genes that are associated with that function? And the answer is gene function enrichment analysis. The kind of enrichment we're talking about here is a relatively simple mathematical statement. An enrichment score represents the fraction of genes in your list, the experimentally determined list, compared with the fraction of genes in the whole genome that have that specific function. And you can repeat this analysis for as many different functions as you like. You know, is function A enriched? Is function B enriched? Is function C enriched? And then once you've said, okay, this function is enriched, it's at a higher ratio in my list than in the genome as a whole, you have to follow up and say, is this a statistically significant difference? Or is it really that I've tested so many functions that just by chance, this one has come up as enriched? It's necessary that the genes in your list come from a single genome and that you have geo annotations for that genome. In the VEuPathDB, we've associated geo annotations with all of the genomes. The statistical test is based on Fisher's exact test. I have a little quote here. Use Fisher's exact test of independence when you have two nominal variables and you want to see whether the proportions of one variable are different depending on the value of the other variable, and use it when the sample size is small. In the case of gene enrichment, we're testing if the fraction of genes with a particular function in a gene list is significantly different than the fraction in the whole genome. A little example, if 6 of 10 genes in your list have function A, you would say how often, meaning how unlikely is it that 6 of 10 are found for function A if you just randomly draw 10 genes from the genome. And you're testing against the null hypothesis that the relative proportions are not different. This is equivalent to the hypergeometric test. And I'm not going to go through the math there, but a different equivalent version of the math. Oops. Yeah, so I'm going to make one more analogy and then go through the Fisher's exact math. I play poker, so I like to think about statistics in this way. If you're dealt 5 cards from a standard deck, how likely is it that you'll have 2 aces? So your hand has the function ace for 2 of the 5 cards. So 0.04 is your ratio. The deck has 4 aces, so 4 out of 52, so 0.769 is the fraction of the deck that is aces. So your enrichment in your hand is 5.2-fold. But to compute whether this is statistically significant, you first need to know the exact probability to get this hand. So you have to compute all possible combinations of 52 cards in a 5-card hand, and then compute what fraction of those contain 2 aces, and that'll give you your probability. That's expressed in Fisher's exact test this way, where A, B, C, and D are frequencies, and N is the total frequency. A are the genes that have function A in your list. B are the genes in the first list and not the second, meaning genes in your list that don't have that function. Genes, C is genes in the second list, so genes of function A that are not in your list, and D is the rest of the genome. So it's just a mathematical expression for Fisher's exact test. It's not something you tend to work out by hand in your calculator, since we have the statistics built into the tools. Now, if you make this test for a particular function, you're likely to go again and test each of the different functions in the gene ontology to see which one might be enriched in your list. So you might hit one just by chance. In other words, you've done so many tests and each one at the same p-value that it's especially true that you might get one that's rare, meaning if one time you draw a list or you look for a function and that function is pretty rare in the genome and it shows up in your list, that might give you a significant p-value. You have to correct your statistical test for the fact that you're doing it over and over again. And you can correct either by Bonferroni, which is a very strict test correction. You multiply by the number of tests, or the Benjamini-Hochberg false discovery rate, which is a bit less strict. And these give an adjusted p-value, which will always be less significant than the original p-value. But for this kind of analysis, it's correct to use the adjusted p-value. Also, you might want to reduce the number of tests that you're doing. The gene ontology has a lot of terms. Some might not be relevant for your experiment. Some might not be relevant for your organism. There are a number of what's called GoSlim Sets, subsets of the whole ontology that have been organized in a specific way. These might represent targeted groups of function for a specific organism or for a specific type of metabolism, like drug targets. So the slim set may also avoid the lowest, most specific terms in the ontology, and therefore have fewer total number of terms that need to be tested. So there's a pre-built set of GoSlims in the Gene Ontology Consortium, but it's also possible to make a custom set of terms that are the ones that you want to test, and that would be a custom GoSlim set. Once you've got your enrichment results, meaning a set of terms that are enriched in your gene list, it's necessary to go a little further and try and interpret what is the biological meaning of these terms. Is this set of functions relevant to the process, the experiment you used to construct your list? Are the p-values strong enough to support an argument for the importance of this function in this experiment? If your p-values aren't strong enough, can a larger or smaller gene list be used, perhaps at a higher or lower fold change? Can a more restricted GoSlim be used to emphasize relevant functions? Sometimes the enriched terms are more general or more specific than the terms that are often commonly used in the community specializing in your organism. So it might be helpful to generalize across a group of related terms and find what they have in common. The geo terms can be clustered by meaning. There's a set of relationships among the terms, both parent and child, and related to the same general function. And there's a few different ways of making a word cloud, clustering the ontology terms by how they relate to each other. A cluster of related terms can suggest an important process that's going on. And sometimes some of the terms in a cluster may have a stronger p-value, which suggests that that cluster is important. And if you had only looked at the less significant terms, then you wouldn't realize that the p-value probably applies to them all. This particular word cloud is made by a tool called Revigo, which is available in the VEuPath tools. It's also possible to use metabolism itself as a set of terms for enrichment clustering. You can take each chunk of metabolism for a particular function and assign that to a set of ontology terms. We can get these terms from databases such as KEGG, Reactome, and Biopsych. So each pathway is a function term, so all the genes or proteins in that pathway form a set of terms. And then again, you can calculate the proportion of genes in a list that are from a given pathway compared to the genes in that pathway for the whole genome. And again, this can be repeated for as many different pathways or the full set of metabolic pathways, each one being a geo term. If an enriched pathway is found, you can generate a nice figure by highlighting the genes in your list on the pathway map, something that looks like this. So I'm going to finish up my part just by pointing out some strengths and weaknesses of the gene enrichment approach. So the enrichment can find functional patterns in a list that's too large to fully understand just by reading down the list of gene names and descriptions. The enrichment method is flexible for your choice of functional gene sets. However, it does require a well-annotated genome. It doesn't work well for extremely large or very small gene lists. If your gene list is half the genome, then you're not going to be getting statistically significant enrichments. And if your gene list has just 1 or 2 genes, then it's probably likely you're going to miss important functions from your experiment. Enrichment is not very sensitive for small changes in a large number of genes from a functional category or pathway. And of course, the enrichment relies on how you've built your gene list. Usually the gene list is constructed with a specific hard cutoff, which is somewhat arbitrary. P-value 0.05, fold change of 2, something like that. So some genes are going to be included and left out of any given gene list. So you may have to go back and try and build a different gene list with a different functional cutoff to see if you don't get what you were hoping for. All right, I'm going to stop sharing and pass it back to Evelina.
All right, thank you very much, Stuart. I'll make myself a presenter. Okay, please let me know if you do not see my screen. I'm on FungiDB webpage now. Right, so what I'll do in the next a few moments, I will show a few demos about how to use enrichment tools within FungiDB and also VectorBase. The cool thing about all genomic sites within the VEuPathDB database is that any of the searches that I'll show you in FungiDB or VectorBase are applicable to other genomics sites. Whether you're working on Plasmodium or Giardia, the logic of how you would find the searches and deploy them is essentially the same. So as you may notice, I'm on FungiDB landing page, and what I would like to do is to create a RNA-seq search using the RNA-seq evidence that has been integrated in the database. And in my particular case, I'm interested, let's say I'm a researcher that is interested in drug resistance in fungi, and I searched for a caspofungin, one of the anti-fungal drugs, and there is a interesting data set in Aspergillus fumigatus that I will investigate using the fold change RNA-seq search. And so because I have pre-run this experiment previously before webinar, you notice that my selections are already highlighted. And so what I'm interested in doing, essentially using wild type grown in the absence of the drug as my control, and then a wild type in the presence of the Caspar function as a comparison sample. And I'm interested in the genes that are upregulated by fourfold. And so I'll go ahead and click answer and wait for my screen to refresh with the results. So I guess you may be wondering why specifically this data set, or why caspofungin is interesting. Caspofungin is a water-soluble drug that has activity against Candida, Aspergillus, and Cryptococcus species, and it's quite often a second line of defense after the initial set of treatments have failed. In some Aspergillus fumigatus strains, caspofungin treatments at high doses have induced an increased fungal growth, also a phenomenon called caspofungin paradoxical effect, or CPE. And so what I would like to know from this particular data set is which genes respond to caspofungin treatment as upregulating their expression. And I also want to see if I can find some interesting genes using enrichment analysis. All right, so my search strategy returned 621 genes, which are listed in the results table below. Now I'll just make it just a tiny bit smaller so it's visible a little bit better. And you could browse through genes if you want just to see what's in the list. You can download these results or view additional columns. But I'll navigate to the Analyze Results tab. And under this section, we have several ways to enrich the data using either Go Ontology enrichment, metabolic pathway enrichment, or word enrichment. And we'll start with Go Enrichment Analysis. For this particular data set, I will use one of the 3 ontologies. And first, I will select the cellular component Go ontology. And this ontology describes subcellular structures and macromolecular complexes to run my enrichment analysis on. Now, because our results are in the background of Aspergillus fumigatus, this is the reference genome that is given to you in terms of the running the analysis. And notice that the search automatically picks up the identity of the organism, so that will be the only option available at this time. And I will leave the rest of the criteria at default, looking to investigate sort of any terms that are either computed or curated. I will not limit to GoSlim terms, as I would like to have a little bit more granular view, and I will leave my p-value cutoff at the default levels of 0.05. Now, while I'm waiting for the search to complete, what I'm actually expecting to see now is the statistics and also the outcome of my enrichment analysis available as a results table. So here it is. The table consists of several columns. You have first Go ID associated with a specific Go term. You also have the number of genes in the background of this term versus the genes that are specifically in your result with this term. And we also have a few columns associated with sort of statistics and percentage of expression of your results, such as % of background genes in your results, and also fold enrichment, which is comparing to the background. There's a column that's called odds ratio. Normally anything above 1, I think, is pretty good, and below 1 may not have any association. And so a large value indicates that it's way more likely for a gene that is in a given Go term to be significant than not. And then we also have several columns that reflect the p-value from the exact Fisher's test and also the adjusted p-values from FDR, file discovery rate analysis, and Bonferroni adjusted p-value. So remember, as Stuart has mentioned, when you work with large samples, it's always good idea to keep your eye on the corrected values. Especially this becomes important for sort of edge cases where p-value may be really close to 0.01, but if you look at the corrected values, they may indicate that perhaps your results are not significant. All right. So in terms of how to perform Go enrichment analysis, that would be approach that you would take. Notice that we have at this moment run enrichment analysis using only one of the ontologies. So if you would like to change, let's say, you know what, I actually would like to see what I may find in the molecular function enrichment. You can do that as well, but only one type of enrichment can be run per each sort of Go or per each analysis. So let's go ahead and run molecular function enrichment by using the same strategy. I will leave all other criteria at default and submit my parameters to the search. So here we are. Now the screen has refreshed and we essentially have a very similar view, only this time we're performing enrichment on molecular function. And so as I would expect from cells that have been treated with caspofungin, you would expect that there's a lot of transporters may be activated, that maybe some enzymes are involved in, let's say, oxidative stress, etc. But how can I actually visualize this a little bit better other than a huge table of Go IDs and Go terms? Well, we have a very simple and sort of easy way to do this. As Stuart mentioned, you can do this in ReviGo. And so you can deploy ReviGo directly from the Go enrichment analysis page. And I already pre-ran this analysis. So the idea that you'll click on the button and the button will redirect you to the ReviGo service. So ReviGo is a web service. It essentially takes in Go terms that we have in our strategy right here. And it also takes in the p-values associated with the results of the exact Fisher test. And once you submit your values to ReviGo, it will run analysis and display them in several different ways. So this is the sort of landing page for the scatter plot, but notice that there are several other options for you available to view your data. If you haven't read ReviGo publications, I highly recommend. It's quite nice to read and learn more about how this analysis works. But I'm right now visualizing the essentially Go terms and p-values that were returned in my molecular function enrichment test. And so what I probably will repeat again after Stuart mentioned that color indicates user-provided p-values that are also listed here at the right top. The size of the bubbles indicates the frequency of the Go terms under the ReviGo algorithm, and that is sort of the Go terms are supplied by the Go database. And the proximity of the circles reflects semantic similarity. So you would expect that all of these genes are sort of related to each other in a way. So if we click, for example, on this group, I'll see that there's a lot of transporters here. And if you look here, there's a lot of reductases, dehydrogenases, etc. All right. Another useful view to look at your results could be a table in ReviGo. And so if you navigate and look at the Go terms that actually went into scatterplot, you will notice that some of the Go terms have been collapsed under a parent. And so what ReviGo does in this case, for example, here's a whole bunch of them that collapsed under the Go for carbohydrate transmembrane transporter activity. So what this means and what ReviGo does, it actually reduces functional redundancy. It takes multiple Go terms, finds a parent, such as this one here, and provides a big picture overview of the Go terms involved. And you have an option to essentially review how it was done and if you want to download the results onto your computer. All right. So at this point, I will return to FungiDB again. And this time we will run a different enrichment analysis using metabolic pathways. So for this, I will deploy a phenotypic search. And so if you haven't used FungiDB or any other VEuPathDB sites, notice that once I navigated, I don't have the main menu of the searches that are normally displayed on the left, such as here. And so that's no problem. All of your searchers are always available for you at the top. So at any point, you can start looking for a new search to deploy. In this case, I'm interested in phenotypes. And I want to look at the phenotypic evidence that is available as curated records associated with disease. So under the phenotypic evidence in VEuPathDB, we have several data sets. That includes data sets from the host pathogen interactions. We also have manually curated data and data integrated from the community projects. And in my search, I will utilize the manually curated data from PHI-based database. So what I'm interested in, and again, because I pre-run the search, my selections are already predetermined, but I could say I want to reset values, and then I'll just come back and look for aspergillosis. So aspergillosis is normally an invasive disease caused by aspergillus, and it's quite difficult to treat. So what I'm interested in this particular search, pull up any genes that have manual curated records associated with this disease, such as maybe they're important for virulence or any other phenotypes associated with disease in animal and human models. And so I will select any possible annotations and run my search to identify these genes. And so once the search refreshes my page, I notice that my criteria, and I can review them really quickly, for 4 different qualifications of the disease returned 159 genes. These genes are summarized in a tabular format underneath the search strategy. And I can further look at the enrichment by navigating to the tab underneath the strategy. So in this particular case, I will run my analysis using the metabolic pathway enrichment tool. And notice that when I arrive to this screen, I may have a drop-down menu actually from several species. So what this simply means that the records that I run my search on were annotated and were collected from several genomes, and that includes several Aspergillus species. You can only run an analysis on one genome at a time. So in this case, I will select Aspergillus fumigatus strain A1163. And I will set up my search to use both sources for annotation, such as CAG and Metasig, that we integrate in VEuPathDB, and leave the rest of the parameters the same, such as EC exact match only, and do not exclude incomplete EC numbers. Now I know we have not really discussed yet what EC number is or how they look like, and I will cover this just in a few seconds after we deploy the search. So I'll go ahead and click submit. Right, and so here's a table with our results, and again it looks somewhat similar to the Go Enrichment Analysis as well. Multiple columns with sort of more granular view in terms of the genes in the background. You have your statistics, and you also have a pathway ID that is applicable to a specific metabolic pathway that has been identified, and pathway name, and a pathway source, such as where these annotations are coming from. Notice that my p-value may be within the 0.05 cutoff. That may be significant, but if you look at the adjusted p-values, they actually changed, and they may not be as exciting as I initially thought. So as a researcher, you should always think about the biological sort of questions that you're asking, the sample sizes that you're working with, how they apply to particular organisms, and ask, does the question make sense? Do you want to accept the result as statistically significant or not, or look at individual cases? Now, I know that when fungal cells are treated, for example, with azoles, or when patients undergo treatment with azoles, certain pathways may be upregulated, and this includes the ergosterol pathway. That is specifically quite often you find mutations within the genes that that allow the pathogenic fungi become resistant to azote treatment. So I'm actually interested if one of these pathways came up within my enrichment analysis. So I'll go ahead and search for ergosterol. And so here we are. We have one hit where one of the genes actually belong to this pathway. So I'll go ahead and click on it and see if I can get more information about this particular pathway. And when we arrive to the FungiDB records on the ergosterol biosynthesis pathway, this pathway is described and visualized within the Cytoscape. It's an interactive interface where you can zoom in, zoom out, look at individual nodes, zoom in, zoom out, and look at the particular enzymes that are placed within this pathway. And now we're looking again sort of at a bunch of numbers, but what these actually are, right? These are EC numbers, and the EC numbers are associated with enzyme commission numbers. This is a database that provides classification for the enzymes currently known. There are 7 main classes of the enzymes, such as listed here. And every single number within the EC number has a meaning. So for example, if we take EC number 1214, one stands that this is enzyme that belongs to the main class of oxidoreductases. 2 number stands for the subclass that normally describes the type of bond or compound that the enzyme is acting upon. We know that within the oxidoreductases, this particular enzyme is acting on the aldehyde or oxo group of donors. Now followed by number 1, this is a subclass that defines further classification of the enzyme and indicates in this particular case that this particular enzyme will require either NAD or NADP as an acceptor. And the final number in EC number stands for a serial number, and that helps to create a unique number to define each enzyme within the classification. So defining it at the end of 4 will allow us to classify this particular gene molecule as aldehyde dehydrogenase that requires NADP as an acceptor. Now, the EC numbers are normally come from either manual annotation or transfer by similarity. For example, if you run a whole genome analysis and you've done your annotation, you notice that this particular gene that you interested in has 90% similarity with another gene in closely related species based on the domain structure, etc. And as such, you're confident to classify this gene as aldehyde dehydrogenase. So this type of annotation can be done by transferring these records by similarity. Now, if you worked and tried to do metabolic enrichment on genes and various genomes, you will notice that not all EC numbers have 4 numbers. Some of them just have 1, and the rest are missing. And that simply means that some of the genes may not be annotated at all, such as, for example, this one, EC1 has been placed within the main class of oxidoreductases, but nothing else have been curated, or there's partial information available. And so these type of, so the enrichment that will use this type of numbers may not be sometimes very valuable, but nevertheless, you have an option to include or exclude them from your search using VEuPathDB enrichment tools. Now coming back to my results, so now that if we look at this particular node, and if we click on it, it will tell us that the EC number reaction is for a particular enzyme. And one other cool thing that you can do within this particular interface is paint enzymes by experiments. So, for example, I know that I found this particular enzyme in enriching my records that came from the phenotypic curated data. So what I want to do is actually to see if I can paint this enzyme based on the caspofungin data that I have analyzed in my first experiment. So here it is. And so I'll click on paint. And so now within the Cytoscape, when the window refreshes, you will notice that now we have a different view. And so what the view is showing you is how this gene is expressed in Aspergillus fumigatus within the 4 different samples that I have looked when running my RNA-seq analysis. And this is quite useful because you can really quickly glance in terms of how individual nodes or particular genes behave within different experiments. Right. And so before I'll switch gears now and move on to a different database. And so now I am in VectorBase, and what I would like to do is actually to show you how to run enrichment analysis based on the list of gene IDs. So as you remember, so far in FungiDB, what we have done, we actually deployed searches on the records that were embedded within FungiDB. These integrated records were evaluated via either RNA-seq analysis or phenotypic searches. But sometimes, as you can imagine, if you analyze data on your computer, sometimes you may have a list of genes, and you simply want to include this list of genes in a search and run GoEnrichment analysis or metabolic enrichment analysis on the set of these particular genes. And so you can do that in VEuPathDB via a specialized search that's called list of IDs. And so if I just click and start searching for a gene, looking for an appropriate search, I'll notice that there's a list of IDs search available here. This is a very simple search. And essentially what it does, it provides an opportunity for you to copy and paste a list of gene IDs that you would like to run the enrichment analysis on. And so this is what I will do. I'll copy and paste a list of IDs and click on Get Answer. And so when the search completes running, I noticed that I have about 244 genes that I have included in this search. And now what I would like to do, I noticed that there's a lot of unspecified products, and I actually want to see if maybe something interesting will come up in the word enrichment analysis. So notice that we have looked at Go, metabolic enrichment, and we haven't looked at the word enrichment yet. So what this particular enrichment does is that it finds words from the product descriptions, and it provides sort of an overview in case if there are terms that you may be particularly interested in. So let's go ahead and run this analysis. So the list of gene IDs that I have chosen to run my search on were from VEuPathDB, and this is why the search automatically picked up this particular genome. And so I'll just leave my search criteria at default and click on Submit. Right, and so the results are now are mentioned sort of under the main search window within the table. And so what I can do is now to see up the words that are occurring within the product description. So you'll see some serines, some proteases. We also see some glycoproteins as fibronectin. And this is sometimes particularly useful if you know that you're working with a set of genes for which there's not very much information available, whether they have not been annotated or they're just unspecified, unstudied set of genes. And sometimes but they may sometimes have a product description information. So this is one way to visualize this data really quickly if you wanted to and take a glimpse on what actually is in your sample, in addition to running GoEnrichment or metabolic enrichment analysis if you like to. Now, just one thing to mention, that every time you run, for example, GoEnrichment analysis or metabolic pathway enrichment analysis, remember that if a particular gene has no annotation, such as has no Go terms associated with it or have no EC numbers curated, they will not come up in the enrichment analysis. So they will be omitted from the results. But there are ways that you can set up search within the VectorBase or any other genomic site to actually sort these genes out and in a group that you can then take a look at them a little bit closer, whether it's looking at the AlphaFold records or just evaluating to see if there's some useful comments from the community, perhaps describing their observations. Right. One more thing that I wanted to mention about this particular search. So my search that I have just shown you was actually part of the public search strategy that was created by Gloria in the past. And we offer quite a few public searches for you to review in case if you have common questions that people have asked in the past. And in this case, the strategy centers around how you can use not only a custom set of G90s, as I have shown you, but how can you look for particular Go terms that may be present in the list of G90s. Notice that this search is not particularly gene enrichment, but this search, such as Go term search, allows you to look up for specific Go terms that may be annotated within the genes in the step 1. So if you would like to review this strategy, navigate to the public strategies within the vector base and select them from the available list. Right. Now what I would like to do, I will just share in the chat some of the relevant links that I have used in my presentation in terms of EC numbers, Revigo, and Go database. And that should appear within the chat section. And can I just please check if there are any questions that we need to answer?
No, not for now, Eve.
Okay, all right, sounds good. Thanks very much, Gloria. Right, so I think we are at the end of the webinar. If you just joined us recently, I want to mention that the webinar will be available as a recording within the webinar section and also will come out as a link within the follow-up email. So, and at this time, I would like to thank you stewards and everyone who supported the webinar in the back and in the back end, and we will adjourn. Thank you very much, everyone.